Hi, my name is Sarah Jansen, and I will explain some background about our research topic, toxic cyanobacteria identification. So, cyanobacteria are found in water sources all over the world. Known as toxic algal blooms, many groups of cyanobacteria are polluting water sources by producing toxins such as microcystins that make them dangerous for humans, livestock, pets, and wildlife. So, the World Health Organization and the Environmental Protection Agency are actively aware of this problem but are unable to effectively clean polluted water sources effectively. This is due to it being extremely difficult to differentiate cyanobacteria on a species level. Typical methods currently used are the study of morphology via microscopy or by using the biochemical features of the organisms. These processes require highly trained specialists within the field and are therefore highly time consuming. Additionally, this method is not always accurate. We're proposing that the use of DNA barcodes will solve this issue. DNA barcodes are made of partial or complete sequences of a gene that is unique to the species you are looking at. Our objective is to create a DNA barcode capable of differentiation at the species level for the following four problematic cyanobacterial genera: Microcystis, Osculatoria, Cylindrospermum, and Anabaena.
Thank you, Sarah. I just want to start off by saying, hi, my name is Vinny Sanchez, and I'll be discussing the objectives of our project and some information concerning the methodology we have used thus far. The overall aim of this research is to develop DNA barcodes for Microcystis, Anabaena, Cylindrospermum, and Osculatoria general, which include the most problematic cyanobacteria species. Currently, we are employing the methodology described by Saba and colleagues for DNA extraction because it included an easier lysis approach that worked on both unicellular and filamentous bacteria, and because it specifically noted that the results were able enough to amplify the internal transcript spacer gene that we are specifically trying to amplify and create the DNA barcodes for. Now in our research process, the primer annealing temperatures were optimized employing temperature gradient PCR. Our next steps involve cleaning the PCR product using exosep, and subsequently we will be setting up DNA sequencing reactions employing forward and reverse primers.
Hello, my name is Lisa Zavala, and I will be sharing how the primers were designed. The primers were designed by downloading internal transcript spacer sequences from GenBank. The DNA sequence alignment was performed using the program Muscle to find any variations among the species. If they were present, the primer was designed using Primer Blast on NCBI. At the bottom are the DNA isolation results, which were obtained for Cylindrospermum, Microcystis, Osculatoria, all of these showing high quality and quantity. As seen in Figure 6, Anabaena had DNA isolation but was lower in quality.
Hi, my name is Natalie Kirkbride, and I'll be covering the latest results we have gathered and the future progress we hope to make. After obtaining ample DNA product of good quality and quantity from our isolations, we conducted temperature gradient PCR. Upon doing so, we have obtained the following results. Figure 7 contains samples of Cylindrospermum and Anabaena. In the top row of Figure 7 are the PCR products from Cylindrospermum. From these results, we determined the most optimal annealing temperature to be 52 degrees Celsius, as it had the brightest band in lane 7. The second row within the figure contains PCR products for Anabaena. A faint band appears in lane 2, where the sample was annealed at 47 degrees Celsius, which we will use as the optimal annealing temperature. Figure 8 contains sample results from Microcystis, aeruginosa, and Osculatoria. In the top row of the figure are Microcystis PCR products. From these results, we determined the most optimal temperature to be 53 degrees Celsius due to lane 8 having the brightest band. The second row of the figure contains PCR products for Osculatoria. A slight band appears in lane 3, which corresponds to 48 degrees Celsius, and has been determined as the optimal annealing temperature for this sample. After obtaining ample PCR product, we have determined that our next move is to clean the sample and sequence it. If the sequence ITS regions show higher interspecific than intraspecific variation and are distinctive for each species, then the barcode development would be concluded as successful. We will also test our barcodes in practical application by using them on water samples sent to us by the EPA that are known to be contaminated with toxic cyanobacteria. Thank you for watching our preview, and we look forward to answering any questions during our designated time slot on Monday from 3 to 4 p.m.